So I'm Jason Key at SBGrid at Harvard Medical School. Thanks for joining today. I'd like to introduce Steve Ludtke. Steve's coming to us from Baylor College of Medicine in Houston. So today he's going to tell us about per particle, per tilt subtomogram averaging pipeline in EMAN2. Steve, are you there?
Yep, I'm here.
Great, go ahead.
All right, thanks so much. Thanks to the SBGrid Consortium for giving me a chance to talk about some of the recent developments we've been making in EMAN2. Last time I gave one of these, I think it was about 2 years ago, and a lot of the stuff I'm going to be telling you about today didn't really exist back at that point in time. Well, there were a few things that had started. Do you need to do anything for your systems check, or are you all good?
Great, looks good.
Okay. All right, so just a quick summary of sort of what's going on in EMAN right now. So we released EMAN 2.31 last month. That's going to be our last release before we make this big Python 3 transition. An unfortunate amount of time this year has been spent doing that Python 2 to Python 3 transition. It takes a lot of work to transition 500,000 lines of code between the two languages like that. So that's I think pretty much on track now. We should finish that up before the new year because Python 2 is becoming deprecated beginning of 2020. So Spire and Sparks are both continue to be distributed with EMAN2. It's a joint software package distribution. So if you have EMAN2 or Sparks or Spire, you have all three distributed together. There was a little bit of a bifurcation, separation between Sparks and Spire over the last 6 to 8 months due to some disagreements on how they wanted to proceed. So Spire and Sparks are more separate products than they used to be, although all three packages are still sort of built on the same core library base. We're also working with Niels Volkmann to begin to incorporate PyCone into the distribution as well for some of his tomography tools. We've been continuing to develop a bunch of single particle reconstruction tools. We've got some new tools based for doing high-speed classification using bispectral invariance, Gaussian mixture models for looking at dynamics, deep learning mechanisms for particle picking and for a bunch of other purposes. So there are a bunch of things there, but that's not what I'm going to be talking about today. So what I will be talking about today is this new pipeline that we've developed for tomography and subtomogram averaging. So this is moving into the field now of in-situ structural biology, which is really sort of a new field. You might have heard Wolfgang Baumeister talking about it as early as 20 years ago. He was sort of introducing this concept and kind of pushing it as a concept. But progress has been sort of very slow until some of the recent developments, which led to single particle analysis really taking off, have now also led to this sort of in-situ structural biology also becoming more possible. The work I'm going to be talking about today was just published about a month ago in Nature Methods, although it's been around and sort of usable for much of the last year. So I'm going to start out with just a few slides for context. I always like to do this because I find that even structural biologists often don't have a really good sense of scale. So this is a light microscopy image of an epithelial cell. You can see the scale bar down there. We're looking at a cell that's about 50 microns across, something like that. You can see the nucleus very clearly in the middle, and you can see other large organelles around inside the cell, but that's really all the level of detail that you can see. Now, keep in mind that if you solve the structure of this cell, if you determine structural information about this cell to about 1 nanometer resolution, meaning you'd have to have nanometer sampling, that cell would be 100,000 cubed voxels. In other words, it would take a petabyte to store all of the information just in that one single cell at that resolution. We're not talking about atomic resolution here. We're just talking about resolution where you could begin to see a little bit of hints of secondary structure. So cells, human cells, are extremely large. So let's look at something a little bit smaller. So here's a cyanobacteria. This is one of the smallest existing cells. It's a prokaryote, not a eukaryote, and it's about the same size as one of the smallest organelles that you could see in the cell we were looking at in the previous slide. So it's about a micron across. So here's an image of an optical microscope, again with some phase contrast, and this is about the best detail you can get in an optical microscope using when you're looking at cells like this. You can sort of tell what shape the cell is, and that's about it. You say, well, what about super resolution microscopy? Well, that's true. If we do super resolution microscopy on these cells, here's an example of an experiment where they labeled the FtsZ ring, which is involved in cellular division. And you can indeed identify where things are. So, we're looking at here where the FtsZ is localized within the cell, but we're not doing structure. We're just doing localization in this situation. So all you can do is localize things that you've specifically labeled. So again, it's not really that much more information than we had before. If we switch to traditional fixed sections with transmission electron microscopy, you can get more detail out. So now you can, for example, see the sort of spiral around the outside. That's the thylakoid membrane lining the outside of the cell. That's where the light harvesting complexes live in this bacteria. But again, we can't see very much inside the cell. We may get some hints that there are some big molecules around, but that's really all we get. Now, if we switch to CryoET, or just CryoEM in this case, we can see tremendously more detail. So here's the same sort of cell. This cell was part of a phage infection study. So what we're seeing here are virus particles, phages, which are in the process of infecting the bacteria. Inside, you can see some more of these phages floating around. These were phages that were produced by the bacteria. You can see the carbon fixation complexes in here. You can see the thylakoid membranes. This is late-stage infection, so there's a hole over here, and these are ribosomes leaking out. There's a sort of a hole up here, and what you're seeing out here is the thylakoid membranes sort of blooding out of the cell and that sort of thing. So we can see tremendously more detail. The idea of what we'd like to be able to do is look at cells and get into them and say, pull out all the ribosomes and look at the structure of the ribosomes as they exist inside the cell. Or look at the organization, how are the ribosomes clustering in different ways inside the cell, along with having structural information. So it's not just localization. It's true structure. All right, so let me switch gears a little bit and give you sort of one specific example just as sort of a talking point to talk about the technology. What I'm going to be talking about is drug efflux pumps. So these are tripartite complexes in gram-negative bacteria. There are similar sorts of constructs in gram-positive bacteria, but the structure is a little different in different sorts of cell types. In most gram-negative bacteria, you find complexes like this. There are 3 molecular components. One of them sits in the inner membrane, one sits in the outer membrane, and the other one spans the periplasm and then sort of connects the two together. The particular complex we're going to be talking about today is a Toll-C complex, and forms a complex like this. Crystal structures have been solved for the 3 components individually. And 5 or 6 years ago, the model that existed for how these things would form this assembly, so there was no structure of the overall assembly, just the structures of the individual pieces, was that these would form a 1-to-1-to-1 mixture in the cell, and basically the ACRA act to sort of anchor the 2 molecules to each other. So this was the picture that existed back then. So collaboration between Ben Luisi and Hua Chu and Zhao Wang, who's my current collaborator on this project right now, back in 2014, they published a Nature paper sort of solving, by single particle reconstruction, the structure of the complete assembly at low resolution. But then they followed that up with another paper, a slightly different mix of investigators, in eLife in 2017, which got fairly high resolution, about 5 angstrom, something like that, and really nailed down some of the stuff that they saw here. But then earlier this year, Hua sort of moved out of this project now with his move to Stanford. Ben is still involved. Anyway, we have a group of collaborators now, and we published an in-situ structure where the structure was solved directly within cells. So this was the single particle reconstruction, the first published one. Again, it was only at 16 angstrom resolution, but that was enough resolution to identify that it wasn't actually a 1-to-1-to-1 complex, it was a 1-to-2-to-1 complex. So there were multiple copies of this ACRA, and the ACRA actually played a structural role in the formation of the complex. It wasn't Toll-C bound directly to ACRB. ACRA actually formed this sort of sealed ring that connected the two. So the original structural picture had been incorrect. but with the CryoEM, even at low resolution, you could sort of solve that problem. But even once we got a higher resolution structure with the overall assembly, this still had some issues with it, right? I mean, this is spanning two membranes. There's an outer membrane, there's an inner membrane, and then you've got you know, this region in the middle, which is hardly empty. You know, so what happens when you actually look at it in a cell? Is it really forming the same sort of complex as it does when it's detergent soluble? So this is a E. coli, which is overexpressing all of the three components of the project. It says unpublished here. This actually was published now a few months ago. That was that third publication in the list before. And you can see here these little sort of specks that are floating around there. Those are sort of the complexes from the end-on view. And you can see here, these are sort of side views. So these are the two membranes, and you can sort of see things sort of spanning the membrane. Now, you say, well, these are just black specks. How do you know what these things are? Well, we've done a bunch of different ways. You do control studies. If you don't overexpress these complexes, then the black specks vanish, and other things as well. I'm showing this to you in 2D, but this is actually a three-dimensional structure. So this allows you to solve the structure using subtomogram averaging. I'm going to talk more about this in a minute. I'm just trying to give you an idea of why we want to do this first. And when we did this from the cell, if you average together all of the particles in 3D, you get an assembly that looks very much like the low-resolution single particle reconstruction we did before. If you subclassify them, however, you then discover that a large fraction, not quite half, of the particles are actually missing the top member of the complex, like this. So then we have this picture now that the ACR-B sits where we expected, down in the bottom membrane. And the ACR-A spans the gap between the inner membrane and the peptidoglycan layer. And then the toll-C sort of spans across from the outer membrane into the top side of that peptidoglycan layer. So we can actually see in the context of the cell what's really going on, and then we can see that sometimes it seems like that this top component, the toll-C component, is the component which it must be the last one to assemble in place, and it may be that that's related to activation of the channel, that sort of thing. This is an active channel, so you don't want it operating in situations where a drug doesn't need to be pumped out of the cell. Anyway, so we solved this. This is at relatively low resolution, but it was enough to sort of answer an interesting biological question. So how do you go about doing this sort of thing? All right, I'm going to assume that you're all at least a little bit familiar with tomography, but I'll just go through sort of a quick introduction. The idea is we take a cell, we put it into the electron microscope. The cell was vitrified, so frozen in place just like it would be with a single particle specimen. If the cell's really big, you may have to use a high-pressure freezer. You may have to use a fib mill to cut out a lamella, so you're just looking at part of the cell. There are all sorts of fine points. Basic idea, though, is you have a cell that's sitting on a grid. You put it into the microscope, and then you tilt the cell in the microscope to different angular orientations, and that gives you a tomographic data set. Since you know the orientations of all the slices, you can then reconstruct the slices. We do this typically in the method I'm going to talk about today in Fourier space. So you can insert the slices into an empty Fourier volume and then get a three-dimensional real space reconstruction. Now, due to limitations in the tilt geometry, because the grid is sort of flat when it's sitting in the microscope, we can't rotate it all the way up to 90 degrees. We can't look at the grid edge on. So there's a tilt limit. We can only really go up to about 60 degrees before what we're looking through becomes too thick. And what that means is we're missing this sort of wedge of information when we're doing the 3D reconstruction, and that has various side effects on the three-dimensional structure. But we do get a three-dimensional structure of the entire cell or whatever object we've got frozen on the grid. So this is sort of what that missing wedge artifact looks like. So if we look at an XY projection, this is the untilted view, basically, of the particle. You can see this is a individual virus particle. It looks very nice. If you look at it, though, from the side, where this missing wedge exists, so down here, this is Fourier space, this is real space. So this looks pretty nice, but over here, you can see with this missing wedge, things get all sort of smeared out, and the information content is much. Things aren't quite as easy as I just sort of made that seem. there's a whole bunch of steps that are involved in actually achieving these 3D reconstructions. And historically, doing this process involved piecing together multiple sets of software. So David Mastinardi's IMOD package was terrific at sort of doing the original reconstructions of the particles. You could pick out fiducial markers, and you could align the fiducial markers, and you could get a three-dimensional reconstruction. But then if you wanted to go in and pull out the individual particles and then do subtomogram averaging on them and try to push to high resolution, you had to move to other software packages for that. So let's just look at the sort of whole pipeline. So first thing we have to do is we have to do the tilt series alignment. When we collect the individual images, we have the tilt series roughly aligned, but we need to know in sort of 3 dimensions where each of these tilts sits with respect to one another, so we can do the 3-dimensional reconstruction. And that requires a very precise sort of pixel level alignment. Historically, this has been done by adding gold fiducials to your specimen before you image it, and then you can align the gold fiducials. The approach I'm going to be talking about today actually works quite well without gold fiducials, just based on stuff that happens to be present in the image. The next thing is specimen thickness problems. The electron beam can really only easily penetrate about a half micron of thickness. So if you're looking at a cell that's 3 or 4 microns thick, then obviously you can't see through the whole thing. And even if you're looking at something that's only half a micron thick, you start running the nonlinear absorption effects, and you no longer have sort of true projection images of the cell anymore. And these are things that you have to take into account in various ways. The next thing you have to deal with is the fact that the volumes that you're trying to reconstruct are 4K by 4K by 1K, for example, pixels. They're quite large. When you do single particle reconstructions, it's considered big when you're doing a 1,000 by 1,000 by 1,000 volume. I mean, say a virus particle is very high resolution. These volumes are just humongous in comparison. And if you look at reconstructions people try to do now with the new large format detectors, people may try to do a 10K by 10K by 4K reconstruction of something. It requires tremendous amounts of memory, and there are all sorts of issues involved with that. Then the next problem is we have to actually find the molecules we're interested in. If we're looking at cells, we need to find all the molecules that are ribosomes or are microtubules or are whatever it is we're trying to find inside the cell, so we can pull them out and then average them together. Then we have the missing wedge problem, which I already mentioned. That can also have an impact when you're trying to align 3-dimensional volumes, and it can have an impact on the anisotropy of the final averages that you get out. Then we have the problem that the alignment that we did up here, where we say we want pixel level alignment. Well, if you do a good global alignment, there are other sorts of geometric distortions that can take place during imaging. You can have shrinkage and distortions in the specimen itself as it's getting dosed, which means that even if you have a perfect alignment in one part of the specimen, you may not have a perfect alignment in another part of the specimen. So you need to deal with that issue if you want to start moving towards higher resolution. And then finally, the same sort of problem that you have with single particle analysis, you have to do contrast transfer function correction. But in this case, we have tilted grids in the microscope, so the defocus is varying as you move across the image, and there are all sorts of issues you have to deal with that. So we have implemented this entire pipeline now in EMAN2. You no longer need to start with IMOD and then shift to EMAN2 and then shift to something else. You can do the entire process all within one software package, and it keeps track of all of the metadata properly, so you no longer have to shift information about the tilt series from one package to another and that sort of thing. So we have a brand new fiducial-free tilt series alignment, which works extremely well. We have a tiled Fourier, direct Fourier reconstruction method, which also works very well. It's at least as good as the other back projection-based methods that people use. There's a tilt series CTF estimation and correction, which is completely automatic and also seems to work very nicely in pretty much every case we've tried. This is something that existed previously. I may have even mentioned it briefly when I gave the last thing 2 years ago. This is deep learning-based 3D particle picking. So we use neural networks you train up a neural network to identify features and then extract them. And I'll say a little bit about that again today. We have a new stochastic gradient descent-based initial 3D volume generation. This is kind of equivalent to the initial model generation process in single particle analysis, but in this case we have three-dimensional particles. The only problem with the three-dimensional particles is they have this missing wedge, so we need to kind of get that missing wedge filled in. And it turns out that that process of coming up with an initial wedge-free 3D volume is actually pretty tricky. So we have a nice algorithm which seems to work on that quite robustly. I'll show a couple examples of that. Then you have the traditional iterative subtomogram averaging. This is good to get you down to about 20 to 30 angstrom resolution. Even without CTF correction, you can usually get 20 or 30 angstroms. If you do a little bit of basic CTF correction, you can get down to maybe 15 to 20 angstrom resolution using this technique. But if you want to do high-resolution subtomogram averaging, you really have to start working with per particle per tilt mechanisms. Per particle per tilt, what this means is basically we're doing sort of a hybrid between single particle analysis and subtomogram averaging. Instead of doing a tomographic reconstruction and boxing out a 3D particle, we go back to the original tilt series and we find each tilt image for each individual particle and pull that out. So now instead of having a 3D reconstruction of the particle from the overall tomogram, we have a tilt series for each particle, and then we can align each of the tilts in that tilt series to do a final three-dimensional reconstruction. And this has now been demonstrated. So EMAN is doing this, and EM Clarity, which was developed at Diamond, also has a mechanism for doing this. And you can achieve sub-nanometer resolutions using this technique, as demonstrated in both in the papers for both software packages. I'm going to tell you that we now actually have achieved sub-nanometer resolution from in-situ subtomogram averages using this mechanism, although I can't show you any of those yet today because those aren't published yet. But in multiple cases, we've now seen sub-nanometer resolution inside the cell. So here's an example. I'm just going to sort of take you through the process now. So here's an unaligned tilt series. These are 8K by 8K images which are collected on a D64. You can tell the energy filter on the microscope was having some problems that day. That's what those big black splotches in the middle are from. And I left those there intentionally because I'm showing that the alignment algorithm isn't impacted by them. This example does have gold fiducials present, although not a very high concentration. So this is the unaligned tilt series. We run the automatic alignment tool. There's no picking of fiducials, no nothing. You just run the program, and it identifies landmarks. Now, I don't say fiducials there because the things that it identifies as landmarks may or may not be fiducials. If you have fiducials present, we'll tend to use fiducials as the landmarks. If you don't have any fiducials present, it will usually manage to find things that are high contrast enough to use as markers. When I say usually, I have not yet found a case where it doesn't work, other than really bad tomograms where the tilt series was just collected terribly, where it had like a half frame shift in the middle of the tilt series or something. Basically works everywhere. There have been people who have attempted to do fiducial-free alignments in the past, but they always focused on an approach where they identified sort of landmark structures or features they were going to use for alignment by looking at the 2D tilt series. The key in this approach is that we identify the landmarks we're going to use in 3D rather than in 2D, so we know that the landmarks are well constrained in all three dimensions, and it turns out that doing that and using a highly iterative approach, that you can get something which is very robust. So this is what the aligned tilt series looks like. You can see it's very nice. You'll notice that maybe it doesn't seem quite perfect, like there's some little shifts in some of the frames. It turns out that's not a problem with the alignment. That's actually when you have an out-of-plane tilt. We're treating this as if we have sort of a single tilt axis, which is aligned in the plane of the image. In reality, sometimes you have slight out-of-plane tilts due to the geometry of the microscope and the stage. So what you're seeing there when you see these tiny imperfections in the tilt series are actually the out-of-plane tilts. But we get a very nice reconstruction. This is what the reconstructed fiducial markers look like. And as long as you sort of see this characteristic sort of X pattern, as long as these fiducials look symmetric, you know you've got a pretty nice alignment. And as you can see there, this took about 10 minutes to run on 12 cores on a CPU. If you have a workstation with 32 cores, you can get it to run in just 2 or 3 minutes. And again, this was an 8K by 8K tilt series we were talking about. That's not just the alignment, that's also the 3D reconstruction process. It's all integrated. So the reconstruction is handled using a tiled direct Fourier inversion method, which we, direct Fourier inversion has sort of become the standard in single particle reconstruction over the last 20 years. A lot of the tomographic software still uses methods like filtered back projection or CERT, that sort of thing. Some of these algorithms can take a really long time. In our hands, anyway, this seems to produce reconstructions which are a comparable quality to CERT, but a much smaller fraction of the time, and produces very nice results. So typically, when we do these reconstructions of the whole cellular volumes, we generate downsampled reconstructions, which is only 1K or 2K, because that's good enough resolution to identify all the cellular features that you're interested in. And once you've identified the features, then you go back to the original tilt series, the original tomogram, and you can extract sort of the full resolution molecules that you want to average. Here's just an example of how well the alignment works. This reconstruction was done at Caltech as part of their automated software solution. It uses the IMOD automatic tilt series alignment tool, and you can see it produced nothing very useful. These lines just sort of all over the place. It was a failed reconstruction. Again, we did nothing special. We just ran the automatic tilt series alignment, and this is what the EMAN 3D reconstruction looked like. What we're doing here is going up and down through the tilt series. Through the reconstruction. This is past the tilt series. This is the final volume. Same thing was true in the previous one. This is not a tilt series. This is going up and down through slices of the 3D reconstruction. You can't even tell the cell is there. Here, now you can actually see the cell, and it looks quite nice. Okay, so here's just another example we'll use to talk about the automated tomogram annotation. This is a section of a PC12 neurite. The circle you see around the outside isn't the edge of the cell, it's the edge of the carbon hole that the neurite was sort of spanning. This is a 3D reconstruction of the cell, and you can see there's lots of stuff in there, right? You can see microtubules spanning across the hole. You can see ribosomes floating around in solution. There's everything that's in the cell is basically sitting there floating around. So the first thing we need to do is identify the different molecules that we might be interested in. So we need to do some sort of volume annotation. The traditional approach for doing this sort of volume annotation was actually as users sitting in front of the computer, going through the reconstruction, slice by slice, hand annotating all of the features. These are microtubules, these are synaptic vesicles, these are ribosomes, et cetera, et cetera, et cetera. One tomogram easily takes a man week of annotation time, 40 hours of someone sitting in front of the computer going slice by slice through these tomograms. So this was one of the first problems we decided to sort of tackle in this in-situ subtomogram field. So a couple of years ago, we published this other paper in Nature Methods where we developed a convolutional neural network approach to solving this problem. Now, I'm not going to spend a lot of time talking about this today. I'm not going to go into the details of convolutional neural networks. That's a longer sort of talk. But the basic idea is that you train a neural network to recognize one particular feature that might be present in the three-dimensional volumes in the tomogram. In this case, we're training it to identify microtubules. So you can see here's the input, one input slice from the tomogram. After you pass it through the network, you get this annotated output, which has sort of a local likelihood estimate of each pixel in the 2D slice being part of a microtubule. By combining a bunch of these networks, one network for each feature that you're interested in annotating, so you train one network for ribosomes, one for microtubules, one for vesicles, one for mitochondria, and so on, you can then build up an annotation of the entire cell. Now, in this example, you'll notice that you can see that circle there, which I mentioned was the edge of the carbon film. It isn't a mitochondrial membrane like it seems to be annotated. I left that in there as purpose. Left that in there on purpose. The point is, if you don't train a network to recognize a particular feature, then it will tend to recognize features that it finds as the most similar thing to whatever that feature is. So if you go in and then train another neural network which recognizes the edge of the carbon film, then it can discriminate between the carbon film and the mitochondrial membranes. If you don't train a network to do that, then you may get some sort of false positives. So you have to train a bunch of these different networks. The key is that training each of these networks only takes an annotator about 15 to 30 minutes. And once you've done it, you can apply it to an unlimited number of cells. So if you need to train up 10 or 15 of these networks, it's something you can do in a morning. And once you've done it, you can then annotate 50 or 100 different cells. You don't need to spend 50 to 100 weeks annotating all the cells by hand. So this works pretty well. It's not clear that you want to necessarily use this at the level of differentiating between two objects which are of sort of a similar size and shape. You're not going to differentiate between a ribosome that has a substrate or a ribosome that doesn't have a substrate or something like that. You might be able to, but that's probably not the right level to approach this tool. The level that this tool is used is sort of doing this coarse segmentation of the tomogram into different objects of similar shape and size. Once you've done that, then you move to subtomogram averaging and classification processes that are there where you can actually do discrete classification of the things that you found that are similar into different groups. So now switching back to the TOL-C project, this is just a demonstration from the manual picking tool that you can use after you do automatic segmentation to sort of view the contents of the segmentation results. So in this case, when we trained a neural network to do the segmentation, we trained one network to recognize side views of the TOL-C and another view to recognize top views of the TOL-C. Because of the missing wedge artifacts and because of the long length of these the odd shape of these particles, it worked better if we trained separate networks for the two different features. We also trained on ribosomes inside the cell and trained a separate network on ribosomes outside the cell, which had escaped the cell in some fashion. And we're able to discriminate between those four different groups. And this is just sort of an example of the interface there. This can be used to manually update the results if you need to, or just to sort of view the results of the automatic segment. This is an example of the initial model generation tool where you take, say, all of the particles that look kind of like ribosomes, and you can make an initial 3D model from these. So you can just sort of see it evolving here on the side. Here's an example from our ATP synthase. Here's the example from TOL-C. Here's an example of microtubule doublet. These aren't supposed to be high-resolution structures. These are just supposed to be isotropic initial starting models, which you can then use as alignment references to move from this sort of 70, 80 angstrom resolution that you have here down to sort of the 20 or 30 angstroms you can get with subtomogram. Skip over that again. So once you have done your coarse subtomogram averaging, which just involves averaging the 3D particles, the next thing we need to do now is this high-resolution subtomogram averaging, this per particle per tilt based approach. So the three key aspects of this per particle per tilt approach. As I said, we go back to the original tilt series, and we wind up with a tilt series for each particle. And each of the images in that per particle tilt series can then be handled somewhat independently from one another, but also still somewhat associated with one another. So you have basically an initial geometry for how the alignments should be, but you may need to fine-tune the individual tilt alignments by a few pixels because of the poor relationship between the overall alignment of the tomogram and the local alignment. So basically, we're doing three things. We're refining the 3D alignment parameters for each tilt for each particle. We're eliminating bad tilts for individual particles. So say you had, in one tilt, a fiducial marker wandered its way into the projection of that particular particle. You want to be able to exclude those bad tilts on a per tilt per particle basis. And then finally, we want to do per tilt CTF correction. So the alignment step is basically this issue, right? So if you've got your initial alignment of the whole tomogram, this is just a nice 3D cat that we're looking at in different orientations. It may be that in the different tilts, the cat is a little bit misaligned in sort of 3D parameters. And by improving the 2D alignments here, based on projections of the overall 3D tomogram, you can sort of bring that into register a little bit better. And that's one key step in improving the resolution of the final, quote, subtomogram average. The other key aspect here is this per particle per tilt CTF correction. So we have an automatic CTF determination algorithm which works on tilted images, and basically what it outputs is the mean defocus at the center of an individual tilt image. Okay, so it'll tell you what the tilt is here. since we've boxed out all of our particles, we know where all our particles are in 3 dimensions, and we know the tilt of the of each tilt image in the microscope. So from that, we can pull out the 3-dimensional location with respect to that known defocus here, and this distance will give us a defocus offset for each tilt for each particle. And then we can do truly accurate CTF correction on a per particle basis. And this is what ostensibly would allow you to move down to even 3 or 4 angstrom resolution. So in the original EM Clarity paper, there was one other example. There was actually a publication showing how some of these sorts of corrections could move the subtomogram average in the case of the HIV envelope protein, which is a very specialized case. It's not, it's not exactly in situ, where you could move that down to about 3.2 angstrom resolution using the subtomogram averaging technique. And we've replicated that, but we didn't improve the resolution any further than EM Clarity did, so we didn't focus a whole lot on that particular example because it's so artificial. I can get into that more if people have any questions about it. But anyway, so it is possible using the technique. Mathematically, it's possible to get to near atomic resolution, or, well, not near atomic maybe, but 3 angstrom or 4 angstrom resolution using this technique. The question is, how limited are you going to be if you try and do this in cells? So again, this is an example not in cells. This is an in situ example. So this was a published subtomogram average of a ribosome, which came out using PyTom. I don't know, maybe 10 years ago or something like that, or 6 or 7 years ago or something like that. And he'd achieved 11 angstrom resolution there. This was another example that EM Clarity also used. Here's the reconstruction done using this EMAN pipeline. The data wasn't collected with the sampling of the data collection was sort of limiting the resolution to an extent, so we could really only get this to about 9 angstrom resolution, but we could get a reconstruction which is a little bit better. What we're seeing in the middle here is a single particle reconstruction of the same ribosome, which has been sort of low-pass filtered to about 6 angstrom resolution. So this is sort of the ground truth. You can see here's one of the RNA helices. You can see here the RNA helix in the EMAN reconstruction is better resolved. Here, for example, there's another little feature in here, this little square region here. You can see it here, and you can see how it didn't come through in the original reconstruction. So doing all these corrections can have a positive impact, but if the data wasn't collected with high resolution in mind, that's often still going to be a limiting factor. So that's pretty much the end of what I wanted to tell you about today. Some of these structures where we're beginning to see subnanometer resolution in situ should be emerging over the next few months. Certainly, if you're not working in situ, if you're working with in vitro specimens and you collect your data with the correct tilt series parameters, you should be able to achieve 4 or 5 angstrom resolution now using the subtomogram averaging technique. For typical in vitro problems, the advantage of the subtomogram averaging is simply the fact that if you have a lot of flexibility in the protein or the molecule, something like that, if each particle is in a slightly different conformation, having some three-dimensional information about each particle can be very valuable in trying to sort of subclassify them, group them together, and to figure out what the 3D dynamics looks like. If your target is to get high-resolution structures from purified molecules, single particle reconstruction is obviously still your best bet. So this is my first point here in the future, that people who are collecting tomographic data right now are not necessarily collecting with optimal parameters to do high-resolution subtomogram averaging. The assumption for a lot of this data collection was always that, oh, well, we're never going to get to better than 20 or 30 angstrom resolution anyway, so the parameters were selected with that in mind. So there are things the community needs to explore now, larger tilt steps, using narrower tilt ranges maybe, playing with how the dose is distributed among the different tilts. So there isn't necessarily a right answer to this at this point in time, but there are things the community needs to look at. There are still some issues surrounding movie mode imaging. A typical tilt for one of these micrographs is only going to have 1 or 2 electrons of dose. So now you're talking, if you're taking movie mode imaging, you're talking about taking 1 or 2 electrons per square angstrom and then subdividing that into movie frames. So splitting that up even further means you wind up with these movie frames which have almost nothing in them at all. So it raises the problem of movie alignment to sort of a whole new level. We then have questions about how much interference are we getting from the surrounding cellular material when we extract one of these per particle per tilt series. There are tricks that we use to make this work well, and we were actually somewhat surprised that we were able to get the subnanometer resolution using this approach. It seemed like the interference from the other stuff in the cell would probably be too much to allow that to happen, but it turns out it's not if you deal with it correctly. Then you have the issue, of course, of particle variability in cells. When things are in cells, all sorts of stuff that we don't understand is going to be going on there. I mean, part of the reason you're doing the structure is to better understand what's going on. So now you may have unknown molecules interacting with your molecule of interest. You're not just looking at the stuff that you purified anymore. You're seeing what's actually happening. So that raises a lot of questions. The level of variability in the cell is obviously going to be massively higher than it is outside the cell in purified specimens. And then finally, if you're looking at something that's thick, then you need to have some approach for making it thinner. And pretty much the only viable approach nowadays for really doing that is using these cryo-fib mills to mill lamella out of the middle of your cell, so you can look at least one thin layer of the cell at high resolution. But of course, that suffers from all sorts of experimental difficulties itself. It's the method of choice if you're looking at eukaryotic cells, but it's certainly not sort of a plug-and-play solution at this point. So with that, I will acknowledge the people that were involved in the project. I already sort of acknowledged most of the TOL-C project collaborators, and actually I didn't talk about the nuclear core complex today. Then back to my title slide, and I'll take any questions people have.
Great. Thank you very much, Steve. If you have questions, you can send them to me by chat. If you've got a mic, just say that in the chat. I could unmute you. I've got a question already. Is there a minimum amount of total dose required for per particle CTF fraction?
The per particle CTF seems to work okay with around 1 electron per frame, 1 electron per square angstrom per frame. You might be able to get away with a half an electron per square angstrom per frame. Typical tomograms that people have collected on cells have used much higher doses, and the whole tilt series may include 100 or 120 electrons. But if you're going to be targeting high resolution, obviously you have to limit the dose to much lower levels. Either that, or you're only going to use a very limited portion of the tilt series. So I would say around half to 1 electron per square angstrom.
I have a question, maybe a little naive from the annotated neural network models for the cells. How generalizable are those? Are they shareable at all? Or if you have the same detector and you're working on a similar system, can you take advantage of someone else's hard work to bootstrap your own?
That's a very common question, and there are other people who are working on these deep learning-based tomogram annotation tools now as well. Although it's a very challenging area because the tomograms have so much data in them that there's scaling issues still. I would say we don't have enough results from enough different types of cells to have a really good answer to that question yet. Ideally, yes, you'd just like to have one trained network or one set of trained networks, and you could just apply them to any cell that you wanted, and it would at least handle, sort of say microtubules or something like that. I would say right now we're not at that point. What the neural network is learning includes some features of the texture of that particular type of cell. So within a particular type of cell, I'd say it's okay. So if I collect 100 tomograms on the same type of cell and a similar microscope, you can probably apply those networks. But if you try and apply it to a completely different type of cell, my guess is it's not going to work all that well, at least not without a little bit of retraining.
Great. So working with other investigators who've been doing tomography, I've seen a lot of the struggle that they've gone through is what you hinted at the beginning of your talk, so jumping between different software and trying to make a pipeline out of. So to see all of this implemented all in one sort of continuous shot in EMAN2 is pretty exciting. Where do you see it going from here?
I think really the next step right now is actually optimizing some of the data collection protocols. I mean, we're continuing to develop the software. I mean, classification is obviously a big point that still needs further development. We do have some tools in there already for doing 3D classification, and they work. But do they work well enough to identify 50 different states of a ribosome? I don't know. I don't think we have enough data or enough experience to sort of answer some of those questions. So I think there's still a fair bit of work to be done. Obviously, once we sort of can achieve sort of 8 angstrom resolution routinely from cells, then obviously the next step is going to be to push down to sort of 5 or 6 angstrom resolution in cells. And I think that's going to take even further modifications. So there's lots of work still to be done.
Great. Any other questions? Great. Well, Steve, thank you very much. I know that I've seen people really struggle with processing their tomography data, and the development.
I would encourage people to at least try, just give the tilt series alignment a try. It takes almost no work to set up. You just sort of throw your tilt series in there. There's almost no parameters to give it, and just see how it works, because we've had a really hard time finding failure cases for it. So give it a shot.
It's a nice place to be in.
All right.
With that, we'll wrap it up. Thanks again, and everybody, thank you for joining. Take care.